few pairs of legs are got all these little hairs on them, and they're spread out like a net to capture stuff.
That's cool. Is Paragorgia the one with the yellow base?
Yes.
Do you want me to zoom out, Chick?
Oh yeah, you can zoom out.
Okay.
And we're just at the base of this little feature. It's already so interesting.
Mm-hmm.
Yeah.
Yeah.
And we made a lot of great headway.
Yeah. Did they started at waypoint 1?
Well, we moved waypoint 1.
Oh,
Yeah.
okay. So it's not what's in the dive plan.
Right. Yeah.
Okay, yeah, because that would have been a lot of headway.
Yeah, no.
Yeah.
We kind of skipped like about 500 meters of that.
So about like halfway between waypoint 1 and waypoint 2, or
Yeah.
Yeah.
approximately, yeah. That makes a lot more sense. What are those yellow encrusting things?
Sponges. They're demo sponges.
Ah, just like little ones.
Yep, just little encrusting demo sponges.
That is a very interesting one.
Yeah, it's a cool Fareid. And we don't usually see it branching like
Yeah.
that.
I don't think I've ever seen a branching one on this expedition.
Push in there.
Oh, it's got the Antipatherian on it.
Yeah.
And some hydroids.
Yeah,
And a Sathermetra.
fluffy.
Oh, and there's a cool sponge above it too, the Gymnoridi. So like all those little tubes.
All right, come on.
All right, so if you're joining us, we are currently 927 meters on the bottom of the sea. I would like to welcome our viewers from Malaysia, Serbia, Sweden, Belgium, France, Norway, Canada, Germany, United Kingdom, and United States. And also, got a little shout out to Erin Your mom is watching.
I saw, yeah. She puts it up on the screen in her library,
Oh,
and so she has about 600 kids go through her library a week,
wow.
and they're all little Nautilus fans.
That's so awesome.
Yeah, I'm sure if my mom could figure out how to do that, she would.
Yeah, it's too late for my parents, but they're usually online. Always bugging me like what I had for breakfast, what was for dinner. Just take pictures.
I was gonna say, are you sending pictures?
Yeah, they're just like, you're living the life.
Was that a school in Palau you were just talking to?
Yeah.
Oh, cool.
No, it was well, yeah, school, but it was a university.
Oh, very cool.
Yeah, so I kind of introduced the internship programs available.
Oh, amazing. Well
Yeah.
done.
It was so weird because we get the elementary students and they're so hyped up and raising their hands.
That's
C-star
what my mom teaches is elementary.
Yeah.
It's very different interactions between each
Yeah.
age.
So it was cool because I had elementary first, then high school, and then I had the college. So
Oh, big
it's like
range.
3 different types of presentation
You can push in there.
Yep.
with the same info.
It lost a leg.
Yeah.
I was about to say, is that normal?
Yeah, no, they usually have 5 legs. This is a Henricia.
Alright.
Yep.
That's,
Zoom
And
out.
there's
great.
the little stickopathies too.
yeah.
The little red whip light thing?
Yep.
Stickopathy.
Yep.
Is that a type of black coral as well?
It is, yeah.
Mm.
And you can see some of those black whip corals in diveable depths as well.
Oh, so they're in much shallower.
Black corals have a really wide range that you find them in the ocean. You can find them in super shallow water and all the way to some of the deepest depths.
Yeah, it kind of reminds me like when I was going to elementary school, we had this very big branchy coral like that we used to hang our toothbrush on. So we'd go brush our teeth and then we'll hang our toothbrush on it. And I think they collected it from just the beach. And it was part of our classroom, so.
Oh, cool. Yeah, when we were diving, I saw some really big black corals. They tend to live down deeper or in shaded areas because they can't compete with some of the other corals. But they can grow in these really large trees.
Oh, that's interesting.
Yeah, it's been speculated because they actually where the base when you in scuba diving depths, when you see black corals, where the base are, it's usually always in a crevice or an overhang, but the colonies will eventually right itself and grow towards the light. So it's been speculated that the larvae are negatively phototactic or moving away from light. But yeah, as Megan alluded to, even though we have found zooxanthellae in a few species of black corals, they do not have the numbers that you would find in the shallow water scleractinian corals, so they're in full sunlight, they would not be, they would be outcompeted by things that grow much faster. And
Yeah, I also read that if a black coral loses its zooxanthellae, like it's not very detrimental to the coral, and they can actually uptake new ones later.
I'm sorry, if it loses its what?
The photosynthetic algae that it
Oh.
lives with. So down at these depths where we're
I'm
diving,
getting up for
none of the
QCed.
corals have that because there isn't any light down here.
Large dead coral in the middle.
But the shallow water black corals do have some symbiotic algae that live with them.
A dead sponge or something.
yeah,
Or
that sponge. A
It's
fossil.
a weird color.
Yeah.
There's stuff on it. Let's zoom it.
All right. All right. Aaron's back from our temperature check. You can push in now.
It looks like a coconut leaf.
Yeah, so I'm saying it looks.
Uh-huh.
Yeah, it looks like
It's another benthic.
Yeah.
benthic tree pieces, but it's covered
What?
with all these little stars and polychaetes and squat lobster. There's lots of food here.
There must have been some kind of storm that pushed a lot of this terrestrial debris out here or something like that.
Yeah, must have.
Some of these things can float a while and then
Yeah,
get waterlogged and sink.
and we're not too far from the island, so
Yeah, so you're not traveling too far away.
All right. Come on.
Cool.
Yeah, almost exactly 30 nautical miles north of Toby Island. But Toby Island doesn't have much vegetation, does it? It's a,
no, it does.
it does? Okay.
Island Reef doesn't have
Yeah,
much.
yeah.
Yeah.
Yeah, that's
Yeah,
what I
Toby.
meant, sorry.
Yeah.
Yeah.
Hato Bay has vegetation and people.
Yeah.
And what's really crazy about their island is they have what do you call boundaries? Sakay.
The barrier reefs or the...
No. Sakay cement for your house.
Oh, yeah, yeah. The fences?
Not really fences. Like their land, the way it's split up, it's like into small, really small squares.
I missed it on Toby Island, are you talking about?
Yeah.
Okay.
Push in there.
Why is that?
I don't know. I found it very interesting when I visited because it was like it's like from me to you, and then that's one person owns it in like a square, and then it's another. It's just that size, and it's very small.
That's
small
great,
pieces of
Jake.
land.
Thank you.
Yep.
Do you want all the way up?
All right. Come on.
Yep.
What do you think that is? Maybe it was something...
Which thing?
It was like a star. It was right next to the
Spot lobster, yeah.
spot lobster.
Yeah, these are big sponges and dead and live material.
Wow, this is...
Wow.
They're kind of forming like a little reef.
It's
Yeah.
really beautiful.
Yeah, this is really nice.
So these are all sponges, the white
Yeah,
ones?
the white is all sponge.
It's really cool. Is the rubble kind of below the white, just dead sponge of the same?
Yeah,
Yeah.
that brown is the dead sponge.
So just as a time check here, there was a question in the chat. So we have approximately 7 hours and 30 minutes thereabouts of bottom time. So about 8 hours more of dive time. So we'll try to cover some ground here, and if we make it to their final waypoint, I'll be at a depth about 700 meters. So we still have about 200 meters of vertical relief to climb, but more importantly, probably a kilometer and some change to our final waypoint.
Little fish. Oh.
Nice polyopogon sponge.
It's actually Pheronemoides.
Oh. And
But it's in the same family, the Pheronematidae.
what's the distinguishing character there?
So it kind of has that helmet look, where it's kind of recurved on the back, and it's got the really thick spicules at the base, and that's yeah. So it's a Pheronemoides.
So the Pheronemoides has those thick spines at the base.
Mm-hmm.
Got it.
The polyopicons tend to have more of like a blade-like appearance, where they kind of come up or are more fan-shaped, and they don't do the helmet thing.
Is that a fish? I just went around.
There was a fish.
Yeah, tiny fish.
It's peeking out. There it is. And it's back in.
Yeah. It's just doing fish things.
Yeah.
Yeah, if the fish doesn't cooperate, just a quick snap zoom on this white sponge,
Okay.
please.
All right, you can push in there.
All right, thank you.
It's a demisponge. That's all I got. Demisponges are really hard to ID. And we need more people studying them, that's for sure.
What makes it hard to ID? Is it because it looks kind of dead?
No, not because it looks dead, but like most of what we know about these sponges is about their spicules, which you can't see. And a lot of these haven't been described yet.
Cool.
So we just don't have a good catalog for them, especially down deep. And a lot of sponges can look outwardly the same, but be different.
Once you get close.
Mm-hmm.
Huh.
Yeah, and conversely, you can also have the same thing looking very different in
Oh, absolutely.
different environments, or highly plastic growth forms.
Yeah, like these, you know, big bushy branching sponges. I've never seen them grow this large anywhere else. So this is the first time I've seen it in such abundance, and then at such sizes as well.
Nice.
A little cup coral.
Yeah, just building out our species list here for this dive.
Oh yeah, no, it's a lot. There's a lot.
Do you want to zoom on that?
Yeah, if we can just do a
Actually, it might
snap,
be an anemone.
snap
You
zoom.
can push in there quick. Snap zoom.
Oh yeah, no, it's an anemone.
Anemone.
All right. Thank you.
All right. Come on.
Did we collect on a past dive one of these big sponges? These
The branching ones?
I don't know if we did on this expedition, but I think we have collected one in Palau waters. But not like a big piece, but we do have, I think, some fragments.
from the first one?
I believe so, yeah.
Yeah. Okay, perfect. Yeah. If not, I
But
would
I'd have-
definitely want to collect it because yeah, it's just so common and
Yeah,
yeah.
I'd have to double check. Let me see if
Yeah.
I can figure that out now.
Yeah, I don't.
We didn't collect it on our watch, so.
Yeah.
No, I could just have a
Remember
memory
it,
of seeing
yeah,
it in
of
the
seeing
lab. Yeah, but we've seen a lot of things in the lab, so. It could always be worth collecting one here, even if we got one on a previous expedition.
Yeah, maybe a little bit further up on a bit. Yeah, I don't think we'll run out of them. They're just so common.
Yeah, for sure. Yeah. Oh.
Is that can we do a snap zoom on?
It's Candidella gigantea, I think. It's definitely a primnoid.
Yeah, you can push in there.
Wonderful, thank you. Yeah.
All right.
Yeah, just making sure we get one good zoom at least on every new thing we see. Trying to set up Megan and her colleagues here. up
Yeah,
for-
it's super helpful. These zooms are really helpful.
Another one of those true soft corals that we saw earlier. No need to zoom in, but I don't know if we had a name. Aqua umbridae.
Dendronepthia.
Oh yeah, or some, yeah. It's just very distinctive. So yeah, if you just joined us, welcome to the fifth dive of Lebu's Voyage 2, our second of two back-to-back expeditions to explore in and around the Palau National Marine Sanctuary. These expeditions funded by NOAA's Office of Ocean Exploration and Research as part of the U.S. commitment to support mapping of the Palau National Marine Sanctuary, commitments made at the Our Oceans Conference about 2 years ago. And the Ocean Exploration Trust is very thankful for the support, and also very thankful for all the support from the Palau International Coral Reef Center to help plan and execute these expeditions, working over the last couple years really to help plan this expedition, and now also to execute it with many scientists and educators from the Palau International Coral Reef Center on board. It's been an absolutely wonderful experience, and we have still well over 7 hours to go today, and then hopeful that we'll do at least one more dive here in the Palau National Marine Sanctuary before we have to make our way back here. That's going to be a over 200 nautical mile transit back into port, arriving on Friday. So we'll be doing some more mapping along the way. We've already mapped way over a dozen seamounts within the sanctuary, all kinds of different sizes, and including some that are very shallow and isolated. And also got for the first time to see these. And then, so yeah, this Friday our expedition will unfortunately conclude, but there'll still be one more expedition in Palau aboard EV Nautilus, so that will be a 17-day expedition conducted in partnerships with scientists at the Scripps Institution of Oceanography and the Coral Reef Research Foundation. That expedition will focus on exploring the physical and biological oceanography around the Palauan Islands in order to understand the island mass effect and its impacts and its beneficial impacts on offshore ecosystems, including those found within the Palau National Marine Sanctuary. And so still a lot more to explore here over the next month. Hopefully you can tune in and join us at nautiluslive.org.
Thank you for
There's an actinoscapia. Looks like it's perched on a black coral.
Are these green spots kind of, or is it just the camera playing, or the lights?
The what spots?
Kind of greenish. Are those
Oh,
algae?
no, no, there's definitely no algae. I
Oh,
think
okay.
it's just the blue of the water
Oh,
and the yellow of the sediment
okay.
making it look green. Yeah, it's kind of like a trick of the eye.
Yeah.
Because remember when we broke off that piece of rock,
Yeah.
the inside was like a really rich yellow.
Ah. Cool.
Oh, that's a cool sponge.
Yeah.
If this were truly a hydrothermally active area, then there could be green clays, like the smectites are typically kind of greenish, but alas, no hydrothermal activity here.
No. That was a long shot anyway.
Yeah.
This is a Leporella, and if you take a zoom in, you'll see that inside the sponge there's tons and tons of little anemones.
You can push in.
Those are the Spongiactis anemones.
Oh, wow. That's cool.
There's so many of them.
The texture almost looks like cloth, like linen.
Yeah. Yeah, it's a little bit soft, but not soft enough to wash up with. I like all the little channels, which is what makes it so nice for these anemones that live here. They live inside those little channels.
Oh, so those are anemones?
Yep.
Uh, that's really cool.
Do they ever get bigger?
No, no, the anemones are always that size. They're really small.
Hmm.
This is one of the biggest Leporellas I've seen.
All right, come on.
Yeah, looks like for those that are following it with the dive plan, we're just starting to climb. We're at the base and about midway up this second conical feature within the center of this seamount. So have a little bit more to climb up to the top of this feature, and then there'll be a small descent again, and then we'll be climbing up a very steep wall towards the a rim of the seamount that is kind of very, very sharp edge. We still have good 7 hours of bottom time, so should be able to cover some large ground and keep exploring what's been a really extraordinary benthic environment with a wide diversity and a high abundance of a lot of different invertebrates, including many which are considered indicator species for vulnerable marine ecosystems. Many of these corals and sponges are classified as that by international bodies that regulate fishing in offshore areas. Yeah, really remarkable. Yeah, with that black background, that's beautiful.
The starapathies?
Yeah, I don't recall seeing one in this dive at least.
Do you see a fish down
Oh
in the
yeah,
crack?
there's fish down there.
I was really hoping for an octopus, but
And it's not too late. Still have 40 minutes.
Yes.
Herc-sized crevice.
Yeah.
All right, push it there.
Oh. What? Looks like a blue color.
Yeah, it's like gray.
Hmm.
So what is it, Jake?
Cuskeel.
Yep.
Yeah, go Jake. I like the view on Atlanta.
Yeah, it was cool. The upper camera view is pretty neat, like looking down into the crevice. Getting that fish eye angle.
I'm zooming out.
Roger that.
Yeah, that coral just looked a little different than
The whip,
this.
that one.
All right, can push in there. Get a zoom.
It's damaged a little bit.
Yeah.
Hmm.
Yeah, that's what. All right.
It looks like the polyps are closing upwards. So it's not a Norella. So it's another kind of primnoid coral.
Yeah, thanks, Jake.
Maybe
Okay.
Candidella.
Okay, come on, Aaron.
Wow. Some really interesting
Yeah, this terrain is cool.
terrain, yeah.
So many primnoids too.
Yeah. One of these, if there's any chance, we had an earlier request for a collection of these Anelopsamias, if there is one. It's kind of intact. It's look kind of small. Yeah.
These are hard to collect.
Yeah.
And I'm sure Jake can do it.
Yeah.
Oh yeah.
I think, yeah, but these are kind of small, yeah. But we'll move on, yeah.
Move on.
Yeah,
Oh,
thanks.
you want a bigger one?
Yeah, just a slightly bit, because this is going to fragment, so. Yeah, I'll keep my eyes open if I see a bigger one.
But they don't really get much bigger than this, usually. This is like an average size.
I mean, I think if you can get it at the base, this was be either one of them, like, but maybe this one.
Yeah.
I feel like it'll be hard to get like right at the base
Yeah.
without cracking it.
Yeah.
I don't
Yeah,
know.
It's one of those ones where if you hook it and just kind of lift it, you might be able to grab it without breaking it.
Without cutting it.
Yeah, you
Just sort of
don't
breaking it
want to
off.
close the jaws on it though.
Yeah.
No pressure.
And we don't have these are quite abundant. We can look for if the different areas easier for you to work in.
Okay.
Yeah, because this is like a cliff, kind of.
Sure is.
Yeah, it is very, very steep terrain, and
Look at these. They look like they were shattered.
yeah, these kind of very straight edges. Do you think like cutting one of these pieces of rock there at those raised edges? I mean, I'm gonna guess
Yeah.
this is all sedimentary, but that would be kind of a place to try to collect a rock sample.
Yeah, we could do that. Or
But yeah,
There's also some other big Anelopsami on that rock.
oh
over
yeah, there we go. That's what I was kind
Yeah.
of looking for. Yeah, because if that breaks, it will.
there's several. If you look at there's.
Oh,
in
the
this
other
view.
one up there is not
Look.
an Anelopsamia.
Oh yeah, that's not sure.
But yeah, if this, I don't know if that's too wedged in there. That would be a good size.
So the difference between those two, one is hard, one is soft?
Yes.
Huh.
Yeah, and then if you would really zoom in, the soft one would have 8 polyps with 8 tentacles that are branched, and this one would have tentacles in multiples of 6.
Oh.
And yeah, so they're in the same group, but quite different. And important to know that the term that we use, corals, is actually grouping a lot of things, some of which are not
I'll push in
related
there.
closely at all. It's more a term that we use to describe things that are kind of functionally this shape. And yeah, I think if this one could be as close to the base. It is going to be very brittle, though.
Yeah.
Looks kind of hard to get the claw in there.
Yeah.
Yeah.
Yeah. we can move on, Jake. Yeah,
it's like right between the...
I'll try not to point out others that are wedged in between the crevices. Sorry, I'm 2 for 2 on that one.
It's nice to have a good zoom though.
Yeah.
All right. Come on.
Maybe there will be the perfect one at the
Yes.
top of this little cone,
You never know.
or an octopus.
Oh, there's a fish.
Look, there's another one in a crevice. Look, right there, between
They
the
like the
rocks.
crevices.
They just get harder and harder.
Here's some more of your Gucci corals.
Oh, what's that on the bottom?
What's
Oh, it's a
where?
fish. Yeah.
Oh, an Oreo.
Oreo, yeah.
Yeah, they catch your attention because they're so wavy.
What is the yellow coral?
That's the Enelopsamia.
So striking.
Mm-hmm.
How's the position on that?
Yeah, it's just like a little small. I just
Okay.
fear that
We need
if
a little bigger.
yeah, a little bigger because this is going to be very brittle.
Oh.
What do we? Can we take a quick look over there?
Yeah, I think this is Enelopsamia as well, but it's not the yellow version. So
Yeah.
there's the Enelopsamia rostrata, and then the rostrata amphiloides. The amphiloides is the yellow one.
Pushing there.
Oh yeah, and if you, Daniel, if you take a look in the upper camera, you can see the helmet
Sponge.
really well, like how it has that like little cave situation.
Yeah. Yeah. Thanks for pointing that out.
It's hard to describe, but like when you see it, like, yeah, it's like, oh yeah, it looks
Yeah.
like a helmet.
I mean, is this one something that we can hook from the base, or is it again a little bit
Maybe.
too tricky?
Yeah. All right, we can try it. Come on.
So within the scientific name, Megan, is there a color to kind of address the different type, or
No,
it's
so
just the
we
name?
use the amphiloides to designate the yellow one, but it's not, it's more of like uh, just sort of a morphotype
Ah,
designation,
okay.
So you're thinking,
but the species is Enelopsamia rostrata.
so you're thinking closed jaw and just hook it?
Yeah,
Yeah,
that looks better.
yeah, you could just like balance it. Yeah, if we had the parallel jaws, you might be able to do it, but with these ones, it's like really hard to do it with the jaw open.
Falling off. And you push in there.
Just a tad bit. Yep.
Yeah.
Do you think it's... What do you think about
Just
that?
think about it a little bit.
Should
and
we
it
not
breaks.
bother, or...
Yeah.
Yeah, I think that's going to be too brittle in that position. It's
Well, you
just
can always, if you don't mind a small piece, suction sampling it.
yeah, I think we were trying to get a bit more of an intact colony, but yeah,
Yeah.
let's move on. We'll have hopefully something. Sorry, Jake, I keep giving you the hard ones. Yeah.
I think they're all going to be hard.
Yeah.
Judging by how that one just...
Yeah, our slurp is also a bit clogged at the moment.
Oh, is it?
Yeah. So...
Yeah, let's move on. It's going to be too.
Yep. Look at that crab.
Yeah, that's a real crab.
That's a crab.
Oh, it's not sure what to do.
Push in there.
Oh. One, two, three, four, five, six. It's a pretty big crab, huh?
Yeah, they're big crabs.
What, do we know what species of crab this is?
This is Chasion micronasicus.
Huh.
Almost at the top.
That one wasn't nearly as skittish as our other ones.
Yeah, I think we've done really well on this watch. Probably close to.
Oh,
he's climbing
okay.
up. Yeah,
You can
come on up.
Sure, come hang out. Let's go on an adventure.
Very curious.
I think it's trying to fight you.
Yeah.
Sees claws and
It's
thinks
like,
it's
yeah.
another
See, at
crab.
this point, Jake, you could sample it.
Reverse, reverse.
Now when people ask, has anything ever attacked the ROV?
Yeah,
Yeah,
we
it's
got a
like, oh, there
crab.
was this one crab. Yeah, it's not really acting that aggressive.
No.
I actually had that question not too long ago.
Last year at ONC, we had a arm battle with an octopus. A giant Pacific octopus was holding on to the manipulator arm.
Oh, really?
Yeah. It was hiding underneath one of the notes.
Can you imagine how cool that crab would feel if the ROV just took off because it went?
Yeah. He's like, yep, I sure told him.
Yep. So I beat back the devil.
Is that the same?
Oh, there's another fish.
Oh,
Oh,
Wow.
I see the shadow.
that was a big one. Nice big cutthroat eel.
Wow.
Reverse, reverse,
Yeah,
reverse.
yeah, they
Backwards.
do the reverse.
Wow.
Whoa.
It's,
Yeah, this is a
There you go, just-
Snappabranchus.
it's like hits and then it
Yeah.
returns to its... That is
Very curious.
interesting. Mm-hmm. Almost there.
Yeah, indeed. So if my estimates are about right, we've covered about 1 kilometer on this watch. Something on that range. Still a kilometer to go till the very end of the waypoint sequence that we had laid out in the dive
Oh, there's
plan.
another crab.
Yeah.
Sorry.
Oh, no, no. Exciting.
there's a lot of these crabs up here. It's cool. I'm glad we're seeing them.
They are really big, aren't they?
Yeah, they're big.
I'm not going to anthropomorphize you, crab.
And I don't even know if we have a Palawan name for these crabs.
Well, they are tasty.
They're not?
They are.
Oh, they are. Huh.
So the common name is golden crab.
Oh.
So there's not really a word for gold in Palawan.
What about like yellow or orange?
Yellow is pipur.
What's crab in Palau?
There's different types of crabs. So, coconut crab is katat. Rogong is, what is rogong in English?
Crab.
It's also crab,
Yeah.
but it's the...
Rokong is the main root. Oh, no. Rokong. Ang aring rokong daw.
Yeah. Rogong ratmol.
Yeah, crab of the deep. Yeah, I'm guessing because we never had a chance to kind of see them or eat them. We don't really have a name for them. Yeah, we're kind of at the edge, yeah?
Yeah, what do you think about May if we try to like crab,
Break a rock?
yeah.
Yeah.
Just off of one of those edges.
Yeah, one of those little squared off
this is-
bits.
Really interesting terrain.
Oh, that's a lot of crust here on this one.
Yeah, I think the
Yeah.
cracks are just like crust.
Yeah, I think so too. Wishful thinking.
Yeah.
Wow.
Right on the top.
Yep, here we are. This is the top.
Very good timing.
What about that yellow one there?
Oh yeah, that
It's
looks.
not really in a crevice.
Yeah, it's
Not big enough, maybe.
Well, that one's
Yeah, the stem
big.
is.
yeah. I mean,
That one looks kind of good.
Jake.
yeah. I mean, I'm just if it could be kind of popped up at the base, and then you try to get it as intact as possible. But I know these things are just so fragile.
All right, push in there a bit.
You could scoop it with a net.
You don't think that would be potentially worse?
Well, if you knocked it off into the net,
Yeah, if you kind of just use the, I don't know.
I don't know. I've only ever seen this successfully collected without breaking it with parallel draws, which you don't have.
Oh, okay.
Yeah, that's just so fragile.
Or if we had like the scoop.
Yeah, that's what I was thinking. You just like
Yeah.
scooped it in.
No, you're there. I have faith.
Are we still going for this one?
I don't know.
Yeah.
I don't know if I can get
Yeah.
the angle there.
Yeah, it's a
Okay.
weird angle.
Hey, right there.
It just crumbles.
Yeah, I think that's, well, all aboard here was just too fragile.
A valiant effort.
Absolutely. Thank you, Jake. Yeah, it's just so.
What's our range to waypoint 4?
Should be about 500 meters, a little less.
Yeah, 485.
Cool.
Yeah, this next trajectory should be interesting. There'll be a little bit going back down and then flattening out, and then there'll be the steepest climb of the dive.
Mm-hmm. Oh yeah, that's gonna be a nice wall right over here.
Yeah.
Jake, since I pretty much have a wrap in, should I turn to starboard once? Take it out? Or just...
It's fine.
Okay.
6-8, it's fine.
Okay.
And I come back around too, it should.
I got a small question before we get relieved. Are the specimens that break easy, do they grow faster? Or
Do
does
they
it
grow faster?
Yeah, do they just depend on...
No, not necessarily. It's just that their skeletons are made out of different things.
Oh,
So that coral that we were trying to collect, its skeleton is made out of aragonite, the same material that shallow water corals are made out of. So it's just very, very hard, but also quite brittle.
I see. Huh. All right, we got a couple of shout outs. May, sending love from Pantin.
Pantin.
Panton?
Yeah,
Oh.
that's where I live in France. It's
Oh,
near
Panton.
Paris.
Nice. Rosie. And we have Mrs. Bendorf's oceanography class tuning in Westfield High School, Northern Virginia. I'd like to thank all of you guys. We are currently going through watch change. We'll be relieved by the 428 crew slowly making their way in while we go get some rest. Yeah.
Thanks, everyone.
Thank you for tuning in and staying with us. And we hope you continue with the next watch coming up.
Get out of here.
Quinlan, would you pass me the sample log for a second? Thank you.
Ali Yata Bene, good morning from Palau. This is the 4 to 8 watch. We are currently diving an unnamed seamount, not for the first time. The seamount is located approximately 30 nautical miles north of Toby Island. And we are currently at 864 meters. So we'll start with back row for introductions. We'll start with our name, our duties here, and
You know we're going down?
That way.
Yeah, okay.
Look down a little bit more for me, Jaden, so I can see where I'm going.
Copy.
I'll push in a little there if you want, video.
So we've got a carrier crab. Sorry, I'm half asleep. I didn't do introductions yet. Good morning. Hi, everyone.
Yeah.
I'm Brian Kennedy. I
Sorry.
am the chief scientist at the Ocean Discovery League and a visiting researcher at Boston University, and I'm the watch lead.
It's good, thanks. Hold that.
thanks, Brian.
Good morning, everyone. I'm Harold Carlson. I'm a PhD student at the University of Hawaii at Manoa, deep sea ecologist, and in the science chair from 4 to 8.
Thanks. Thank you.
Good morning, everyone. My name is Kinlan Yao. I'm the data logger for the 48 Watch. On shore, I'm an undergraduate student at Palau Community College, majoring in STEM disciplines.
Thanks, Kinlan. All right. Front row, we'll start with Chris.
Hey, I'm Chris Krasnowski. I'm here as a navigator and a mapper.
Good morning. Dan, grateful to be sitting in the Hercules chair. You could zoom out for us, please, video.
Thank you.
Hey, everyone. I'm Jaden. I'm the Atalanta co-pilot for the 48 Watch. On shore, I work as a mechanical engineer at the University of South Florida.
Thanks, Shaden.
Hi everyone, I'm Taya Pollock and I'm your video engineer intern.
Hi, Amber. Good morning. And I'm Kim Etsity. I am sitting in the SciComm seat. I'm an educator, teacher, back on the Navajo Nation.
Come up a bit, Jaden. You're going to have to do a little bit higher delta there. I'm going to basically stay just to the north of you a bit. We'll come sideways down for a while. Okay, video, you can push in there a bit, please.
I'm primarily interested in the yellow thing.
Got it. Okay.
Okay, sorry.
So we've got a anthomastus and some kind of yellow encrusting sponge, and then
push in a bit
coral
more.
whip here. All right, that's good, thank you.
Okay, go wide, thanks.
Okay,
Were they able to sample that brittle coral?
No, I don't. They were not. With the slurp being not exactly functioning, and those Enelopsamias are exceedingly brittle.
Dan, when you get the chance, would you mind scooching the Orion arm?
Right.
So we got I have to admit, I didn't watch most of this. I had the TV on in my room, and I fell asleep watching it, so I didn't watch as much as I had planned to tonight. But this is, from what I have seen in the pass down I got from Daniel, this has been quite a cool dive so far. This is a really nifty shaped feature. It almost looks like the top of it looks like Molokini, if you're familiar with the Hawaiian Islands. It's a kind of famous
if you want, Chris.
snorkeling spot.
Yeah.
It's a half crater
Get us out of
with
here.
this underwater shelf, and very much what the top of this feature looks like.
Bridge nav. Let's do 20 meters.
Or Mount St. Helens.
160.
Okay. And then just go back to sled. Sorry. Oh, okay, yeah, you're right. Sorry. There's so much happening. Oh, God, okay.
Look to your right a little for me, Jane. Thank you.
Thank you.
Should be able to you can come down just a little bit there.
Copy.
So all these white primnoid corals, those look like Narella to you?
Primarily, yeah.
Yeah.
From the capstone data, Narella was the most abundant, or second most abundant, but it was one of the super high abundant genera. We saw all throughout the U.S. Pacific, so I'm not at all surprised to see it the dominant thing here.
Thank you. Oh God, sorry. What am I doing? Oh. I thought I did that. It's my bad.
Brian, when you get a chance, can you go ahead and do a really brief description on today's dive plan for our viewers?
Yeah, so this is our southernmost dive of the expedition. We're down in an area called the Southwest Islands. We're just north of the island of Toby. And like I was saying earlier, this was first, we just mapped it yesterday, and we were all pretty shocked at how it looked like. It looks like a very classic kind of volcanic cone with a caldera in it that has been either had a landslide that ripped off half of it, or it blew out sometime long ago in geologic history. But it's like what you would draw like a very middle school science project shape of a volcano, and then just scoop out one whole side of it. So we landed in the scooped out side, and then we moved up into what would be kind of roughly the caldera type feature, and then climbed a little local high, or a little lava dome type feature, which is what we're on right now. And now we're getting ready to move off of that back into kind of the caldera or crater floor, and then we're going to move up to a very steep and narrow crater rim for the end of the dive. And this, so far, from what I've seen, has been probably the highest diversity and abundance of corals we've seen, certainly highest abundance of corals we've seen so far on this expedition. We've got a pretty good strong surface current here. We're down in a completely different current regime than we were up near the north.
You want me to
closer
keep
to the
moving,
main
Dan, or?
Palawan
Yep, keep
group.
moving.
And so there's
Bridge,
probably a
bridge
lot
nav,
more
number 20.
primary production down here, and there's certainly a lot more surface currents in this area as well.
One more step.
Yeah, and this is almost all these white corals you're seeing are Narella, which is a type of primnoid. A few Stichopathes black corals and Commercially crinoids. Also some interesting branching sponges as well.
Thank you for that, Brian.
Zoom sponge, please.
All right. All right, go ahead, you can push in there a bit.
Got it. Yeah.
All right, that's probably good. Thank you.
Okay, go wait, thanks.
Go wide. Okay.
Let's have a look at this fish.
Here,
I don't know, maybe we
push in on the fish.
might have seen some of these already in the previous watch, but
Push in.
just to get a quick look.
Got it. Thank you.
Some kind of cuskeel. It's got a flat head. That narrows it down a bit. All right, that's a pretty good side view. Thanks.
Moving on. Okay, you can go wide, thanks.
Going wide.
So cuskills, I recently learned, they don't look like much as adults, but their larvae are pelagic, and you can actually see them scuba diving at night in the surface water, and they look absolutely beautiful, like these glass, sort of feathery looking, transparent fish. Yeah, look up a larval cuskeel if you're curious.
Come down a bit, please.
Okay.
Push in there for me.
Pushing in.
Come down some more.
Copy.
That's good. Just enough to let me get around a bit there.
Bamboo.
Yep. All right, that's probably all we need. Thanks.
Moving on, thank you. You can go in.
Go wide. Yeah, sorry. Got confused with
Bridge now.
directions.
Let's do
Maybe
another
don't
30.
So those of you who just tuned in, the
Standing
lasers
by.
that you're seeing are about 10 centimeters.
This rock's got a couple probably Narella whips, got a Stichopathes, Bathopathes, looks like a Hemicorallium, some more of those yellow feather stars.
Yeah, 30 meters.
Was this as much coral as you were expecting to be here?
Yeah, I had high hopes for this dive, and so from what I saw, I've seen so far, it is living up to those hopes. Yeah, I expect, given what we're seeing here, I expect the ridge at waypoint 4 to just be spectacular. At the danger of jinxing us.
So the Exploration Trust was founded in 2008 by Dr. Robert Ballard. Dr. Robert Ballard is he's an explorer and he's known for his discoveries of the RMS Titanic in 1985. So that's who is behind the Exploration Trust. Kinlan, I know that you corrected me a little bit earlier. So we're exploring Toby Island, or near Toby Island. So can you go ahead and give me a brief explanation about how you say it in Palauan language?
Oh, okay.
Yeah.
Well, the full name is Hato'ove, but we just shortened the name to Toby.
Okay.
Yeah.
Thank you.
You're welcome.
Can we take a quick look at this before we move off?
Sure. Can you push in there a bit for me?
Looks like a hydroid or hydrozoan.
No,
Or
can we get tighter?
Here, you go full zoom there.
Thorella,
Yeah.
says Steve.
Yep, that makes more sense to me. But it looks like it's got some hydroids in there too, making it harder to tell. Or maybe it's just got some dead branches. I can't see what those brown. This might just be flocculent, like stuck on it.
Yeah,
All right,
maybe.
Thank you.
I see some zoanthids in there too. Interesting. I think of Thorella as like bushier, but that does make sense.
Look to your left a bit for me, Jaden, up the hill.
Copy.
Harold, how much of the earlier dive did you watch?
I stayed up until they hit bottom and then saw that there was no hydrothermal vent and went to bed. But it was pretty sandy when they landed.
Bridge nav, one more step.
How many hours of mapping did we do in this area? It's like a lot, it feels like.
Yeah, it was 6 or 7, I think.
So this area has not been explored before.
Certainly not at this depth. No, there's been some work in the shallow and I think mesophotic around the islands, but out here in the deep, I think the nearest previous dive is about 100 miles away.
That's amazing.
Yeah, National Geographic put down baited cameras here earlier this year. Or
Oh, that's what you were
on
saying.
Toby, not at this exact seamount.
Yeah, there was a drop camera deployment about 50 miles away in the 1950s, and then there was a the Japanese submersible Shinkai 6500 dove in the kind of the bottom part of the trench, which is about 100 miles away. And then there was some more drop cam, National Geographic drop cam stuff, not on Harold's project, but at a previous deployment a couple years ago, out about 100 miles to the northwest.
That's pretty neat that we were able to get this.
Yeah, and we talked about this on last watch, but the difference between what we know in the satellite mapping and what we see in the multi-beam is just a major difference, and this was another example of, we knew this was a relatively shallow feature, but had no idea of the geologic complexity until we mapped. And I think everyone who walked in the data lab yesterday afternoon to get ready to dive plan made some form of audible gasp or exclamation when they first saw what this feature actually looked like compared to what we were kind of expecting it to look like.
Yeah, I liked your description of a middle school volcano. So cool. It's exactly what it looks like.
So we're in an area called a back arc spreading center, roughly here, where you've got kind of the southern end of the plow trench, which isn't very deep, about 105 nautical miles to our east. And then we've got the Philippine Trough, I don't remember how far away, but a couple hundred miles to our west. It's making this an extremely complex, tectonically complex area. And so sometimes you get back island arc spreading, which is where the trench system basically pulls the crust, in this place the Philippine Basin, and can cause volcanism and hydrothermal vent systems and stuff like that. This is an ultra slow moving area, so we don't really expect to find much active right this moment, but in other geologic time frames when it was more active, we would have had active volcanoes and hydrothermal vents in this area, just like you do in the Mariana back arc. And I'm sure that is roughly what formed this feature, but given how much carbonate we're seeing into it, was pretty near the surface at one point as well.
Nav screen's on a holiday. Thank you.
We got another Oreo fish. These are one of the most common fish we see at these water depths.
down, Jane. Turn on it right now. Thank you.
Bridge nav, 30 meters, 160.
And we are coming down off this feature, which is why it's a little bit slow, because the vehicle's got to back down the hill, making the pilot's life a little bit harder. But we should be off of it into a flat area here in the not too distant future, and then start climbing right back up the crater wall.
Amber, are we able to pull like a 3D image
if you can
for
reach
the
that
viewers?
or not, but
Yeah, no problem.
Say again, 3D image?
Yeah, or a map for the viewers.
You can come down a bit.
Is that possible?
I don't know if we have the Norbit running right now.
Oh, okay.
Yeah, it's running. I think Mike started it up.
Oh, okay. Here, let me
the sponge on the
add
bottom
that
right
to the
is
computer
my
in a
interest.
second.
You want to push in there a bit, video? It's good, thanks.
That's probably good for science, thanks.
Okay, you can go in.
This is definitely a Primnoid dive, or at least this, not maybe the whole dive, but this area is definitely Primnoid dominated.
Those ones are the white ones?
Yep.
Or? Yeah.
Auto purple there.
Oh yeah.
Nav, remind me what computer your Norbit stuff is on. Rockproc?
Rossproc, yeah.
Okay.
And push in on the purple bit for us.
Got
We
it.
like purple.
Hemicorallium, we got a Victorgorgia, and we got an not
That's
so
good,
healthy
thanks.
looking Inalepsamia.
Oh, come on, Hurt.
That's all science needs.
Okay, you can go wide, thanks.
All right, you can have yours back again. All right, so we should have that Norbit on Sat3.
Oh, thank you.
And ROV, I also have the still cams in PC4 if you need that.
Alright. Hey, I can see some uphill behind Jaden. 40 meters away.
Oh, I guess the multi-beam didn't lie. So it looks like we're getting off our little our little cinder cone or here and into the sediment. Have a little sediment crossing.
That's a pretty stark transition.
Yep.
Bridge, bridge, bridge nav. One more step.
Chris is cracking the whip on me over there.
Yeah. Don't you want to play on the ridge?
Yeah.
I didn't figure you'd need much convincing.
Well, we are playing on the ridge here. Just.
Not the good ridge. Look at that ridge.
Oh yeah. Ooh.
Chris has your best interest in mind, Dan.
Always do.
Come down for me, Jade.
Copy.
Come right down. I'm gonna come around the rock here. You're on the flat now. You got some water under you, so you can come down on the deck there for me.
Big bath of Pathies, another nice size Enelapsamia. Can we look at this one, please?
Yeah, let me come back around the other way and turn in my tether here.
Once again, Ed McNichol, if you're watching, thank you for the telestrator.
You'll have to come down to probably single digits there.
Copy.
I just need a quick look at it.
Right. Rock's bigger than I thought.
So I've got a different primnoid over here. This kind of tan color one is also a primnoid.
That should do me there, Jade.
What was that?
That should do me for now. Thanks. Push in there a bit, video.
That one.
Oh, good things. Wait. Stop on the rock here. Okay, you can push in a bit more.
You can see how much current there is. You can see the Narella there like vibrating. Right, some type of corallid. Can we look at the pink coral straight down too while we're here?
Right there. Yeah.
That one.
Push in a bit tighter there. Sorry, I got
All right, that's good enough. Thanks.
porch
That's fine.
there. Okay, you can go in.
Dan, it looks like there's still a little bit of Orion peeking into the still cam. I just noticed.
I might have pushed the wrong button here.
Just a tiny smidge more.
Yeah.
Perfect. Thank you.
Taking the turnout there. Let me come onto you, Jaden, and I'll let you spin around and take the turns out.
Copy.
have to come up a bit first before we do that, though. I'll come right under you.
Here's the first Metallogorgia of our watch.
I think I saw one or two earlier.
You did? Okay.
Oh, there's a fish there. We can have a quick look.
Push in on the fish real quick for me. Okay, I'm getting
Different type of
pulled
cusk
around
eel.
here. I got it. You can go wide.
Small one, huh?
Yep. Thank you.
Can you spin clockwise now?
You mean to get the wrap out?
Yeah.
I gotta go counter.
Really?
Yeah.
Oh, yeah, you do. Okay. Actually, hold what you got there.
Copy.
You can just come look south. I'll take it out when I come under you.
Okay.
You look south and come up. That's good.
Anemone. It's amazing when you see these big-bodied, fleshy-bodied anemones on these little tiny corals and not crushing them. Really an appreciation of how neutrally buoyant these things are.
Pushing a bit there for me. Okay, hold that.
When you get these corals in the lab, they're, skeletons are quite bendy, and so that relatively large anemone on that Thorella is shockingly big for how much strength the skeleton actually has.
I know you probably mentioned this, but what symbiotic relationship do they have?
I don't think we fully know. I'm kind of guessing that this is probably not as good for the coral to have these bigger things. So it's either the coral doesn't care or it's kind of mildly bad.
Yeah.
We will see the tissue abraded away around where smaller anemones attach sometimes, but it doesn't seem to cause widespread death.
I'll wait on the next move. Okay, Chris, let me come over and get the wall there. You might even have to step north a bit. We'll see what happens.
Wait, was that, you want me to wait or you want me to move?
Yes, definitely wait.
Yeah, okay. No, we've been sitting for a while.
All right.
Just to your right, can we look at the two corals, coral and sponge